So good evening, participants. Welcome you all to the second day of our 15 days internship course on bioinformatics. So before going into the session, I would like to give a couple of instructions which I already gave yesterday. So I request all the participants to accept the Google Calendar notification. Some of the participants have not accepted the Google Calendar notification. We have sent the Google Calendar notification to you all to the registered email ID. Kindly check your email inbox using our email ID, our institute email ID. You might have known yesterday I said. Please note this email ID which I am posting in the comment box. So please search the Google Calendar notification using this email ID. So we had sent the Google Calendar notification to your email which you gave during the registration. Go to your email box and your mail will look like this. That is invitation, 15 days internship course and certificate course on bioinformatics. Open this mail and just click this yes. Before you accepting the invitation, it will be like this white. So just click on this yes. So if you select this yes, you can directly join the session without waiting for our approval. Like if you click the link, you can directly join the session. Or otherwise, you have to give unmute you all every time while you are trying to join the session. So I kindly request you to accept the invitation. That's the important thing, because after the session starts, we might be in this page like presentation page. So we could not able to see the Google Meet page, and we could not able to allow you all the time throughout the session. So I kindly request all the participants to accept the invitation. So this one thing, and another thing, we had sent the yesterday's session recordings to you to your WhatsApp. So these recordings and feedback link. So I'm sure that most of the participants have received the recordings and the feedback link. If some of you didn't receive the recordings and this feedback link, kindly save our mobile number in your device. If you save our mobile number in your device, you can receive all these things. If you didn't save, it will not be delivered, because we used to send broadcast messages to you all. So save our institute email ID 9488773165. So kindly save this number as Institute of Innovations in your mobile or any other device. So this is another instruction, and we had sent the confirmation mail to all the participants to your email. I noticed that some participants didn't receive the email. One participant name Shweta Raju. I think there is some spelling mistake in this. If anybody didn't receive the email, kindly contact me by WhatsApp so that I will be correcting the email ID from my side. So one email ID, Shweta Raju, I think I is missing. I'm not sure about that. So contact me through WhatsApp. And another participant email ID, Akarnish. Just a minute, I'll search. So Harnish from St. Joseph's College. So kindly give your correct email ID. And another person, Shweta Raj. So your email ID also, I think there is some mistake in that email ID. So I kindly request all the participants to give your email ID, send your email ID. Thank you. So this is all from me right now. So I will switch off my presentation. Professor, welcome you, Professor. Welcome to this session.
Good evening, Professor.
Yes.
So good evening to all the participants also. So can I start the presentation, Professor?
Sure, Professor. You can start the recording, and as usual, you can manage the session, Professor.
Yeah. Thank you. It's already started recording, so I just share my screen. Participants, is this my PowerPoint visible to you? Yes or no? So kindly interact, like even you can unmute or no answer, or you can chat. Yeah, very good. Yeah, good. Yes, sir. Good. So before going to today's session, okay, we just recap the last session contents. So this is what generally we make. Okay, so to have a discussion of what covered. Okay, even if you are on absent, if you didn't attend the class. So tell me the last session topics, and I need a kind of an interaction. Only the interaction gives you the clarification. Okay, like if you don't understand, even because of you, even others can also can learn from your side. Okay, so because I want much of PG and PhD scholars or participating as a participants. Okay, when compared to the UG students, so only I'm taking fast or somewhat fast. But if you don't understand, you can type the question in the chat box so that I can clarify before going to the next concept. Okay. Yeah. So last session topics. Yeah, come with your answers in the chat box. What exactly did we discuss in the last session? Just we have covered one session, isn't it? Only one session on what? Yeah, participants, if only if you interact, then only I can take you further because you have to create interest on me also to deliver more. And if you don't understand, even I should clarify your doubts. Good. So the first initiated participant is Subhashini Shankar. Very good. So we discussed on cheminformatics. According to Purnashri, we also discussed on immunoinformatics. And according to Parky Lakshmi, so bioinformatics and computational biology, excellent. Okay, and also I found from Divya and Shwarma the same answer, Purnashri, Charmila. And what else did we discuss? Very good. From Swapnil, we are getting another concept that is we are discussed on drug likeness filters. In drug likeness filters, we discussed on Lipinski's rule of five. Okay, if a compound has to be uh, proved as a drug, the compound can also behave as a drug if it satisfies any three rules out of four rules in Lipinski rule of five. That's what we have discussed, isn't it? It's a cheminformatics kind of a tool we have discussed. So that is a method we used in for development of cheminformatics tools. Very good. From Maria, I came to know that we discussed on reverse vaccinology. That is an immunoinformatics, one such approach we use for discovering an epitopic-based vaccine. For discovering an epitopic based vaccine, okay, we can use a reverse vaccinology. You know, the first peptide uh, came to the market from reverse vaccinology is meningo meningococcal polysaccharide vaccine. This is actually used for treatment of meningitis. Okay, so you know because of the computational methods, even so many vaccines and drugs are emerged. So this is one such example. Meningococcal polysaccharide is the first vaccine generated from reverse vaccinology. And I also told you one more example. You remember? I mentioned you something like COVAX. You remember? This is the vaccine, multi-tope vaccine generated from reverse vaccinology used to treat SARS-CoV-2 infection. But I also told the efficacy is much more when compared to Covishield and COVAX and what we use in India. Okay, so one can able to target specifically a patient and even a particular strain of virus if you try to use the reverse vaccinology. Okay, just because it's a specific, we can't guarantee that whatever vaccines we develop in India, which can target the Indian population, can target the American population because they have a different genes, isn't it? So whatever vaccine, if you want to develop a vaccine for India population, we can able to develop, and even we can specifically develop up to the community also. For example, if you are residing in Tamil Nadu and if you are belonging to a Nadar community, so whatever vaccine I develop for, it can be a SARS-CoV-2 vaccine developed for a Nadar community, can effectively work for the Nadar community people. But I can't tell how about the Andhra people, how about the Karnataka people. We can't guarantee, but even we can specifically design the vaccine. Okay, so one such approach is the reverse vaccinology. After that, we are discussed on data life cycle, isn't it? So we got an answer from the participant. So data life cycle we discussed or related to pharmacogenomics data management. That is, if you are testing a drug with a patient and if you want to check whether bioinformatics trials, bioinformatics management is coherently matching with the clinical trials or not. For this, we can use a data life cycle. We just compare the clinical database management and bioinformatics data management, and we can come up with a kind of a conclusions, and we can use the data for the prescription of drugs also. Okay, so it can be used for multi-purposes, especially in US, that is in developed countries like US, they have electronic medical record contents, electronic medical record contents, which we call it as EMR. Like how we have an Aadhaar card, okay, PAN card for each and every Indian citizen, and it is of course a mandatory, isn't it? We have to link with the bank passbooks, etc. But in the developed countries, they have taken an initiative that even each and every citizen has a patient ID card, so that we maintain in electronic medical record contents, from before the birth also, from the mother's home, okay, what is the medicines the mother has taken, and after the birth what are the vaccines and medicines the child has taken. Everything will be stored in this EMR server, okay, electronic medical record server. Like if you go to the doctor, okay, so just by scanning your patient ID card, we can come to know your present illness and the history of illness, and they can prescribe the drugs accurately without any delay. So much good initiatives are there in the developed countries like US. So we have electronic medical records, so which is something associated with the data life cycle. There is a lot to know, study about that. Okay, but the time, whatever we have, the time is very less, isn't it? For each and every hour, I am completing multiple concepts. So I'm just elaborating you more. So if only if interact, then I can provide you more information like this. Okay, good. And now, according to Swapnil, so we also discussed on ADME. That is, can someone tell me the expansion of ADME? Because at the last session, that is on 30th, most probably on 30th of September, you have an MCQ, isn't it? Multiple choice questions. So you may get these kind of a questions. Okay, so but your this assessment is not recorded in your certificate. Just like that, you get a participant certificate. So no worries on that. But it gives you a self-assessment whether how much you understand from these 14 sessions. Got it? Yes. Very good. From Mirunalini, I got an answer. Absorption, distribution, metabolism, excretion. Good. Actually, like ADME, there is also called LADMET. Can you someone tell me what is L and T denotes? ADME, you have mentioned correct. But what is L and T in LADME? I just teach you ADME, but while I am explaining about the ADME tool, I have taken you to the pre-admit. That is a tool called pre-admit, which is used for prediction of toxicity also. And I also showed you a slide, okay, on L. What is L? Liberation. And very good from Rahul and Jyoti Krishna. I got the answer toxicity, T for toxicity. Okay. So these are some of the pharmacokinetic properties. If you want to check the pharmacokinetic properties of the drug, okay, one has to check with the LADMET characteristics. Okay. So this is what we discussed in chemoinformatics. So we have so many chemoinformatics databases and tools. Okay, we have discussed on that. Okay, so just now we have discussed a recapping of last session contents. Now let us begin with a new session on biological databases. So biological databases we're going to see on sequence, structure, genome specific, and other special databases. What are the general sequences we can observe in the biological databases? I already mentioned even on yesterday that if you see the big data in the world, okay, so in which sector we have more big data? Now at present, you find bioinformatics is a sector where you can find more big data. Earlier, when the internet was discovered, okay, 1970s to 80s, okay, this was actually the internet came into origin just because of DARPANET. There is Defense Advanced Research Project Network Agency. DARPANET is from a US defense agency. Just because of US defense agency, they brought a kind of internet. But now, whatever DARPANET, we call this as an internet, isn't it? Just because of the origin of internet in 1980s, so we are finding so much kind of a data deposited in the database. So to know about this kind of a molecular sequences like DNA, RNA, can someone tell me what is GSS and EST? There are plenty of sequences, nearly some 500 sequences are there, 500 types of sequences are there. But I'm giving most important thing what we generally use in our daily purposes. Okay, what is GSS? What is EST? I just mentioned, but if you have an idea, very good. For EST, I got answer like express sequence tags. This is what we observe from microarrays, isn't it? In microarrays, we get a kind of an express sequence tag, so nothing but an mRNA sequence, which is converted to cDNA because of reverse transcriptase enzyme. The sample what we use in mRNA. So that's what we call as an express sequence tags. And if you see yes, very good. So GS is nothing but genome survey sequence. Your answers are right. Good. So actually both GSS is just mapping the genome. These are the sequences which going to map the genome. Of course, EST is also map the genome and GSS, of course. And if you see genome, I think you know what is protein, what is gene. What is the genome? Something new. But if you are in bioinformatics, you might have heard about this. Can someone define me what is genome? What is exactly a genome? And how the genome is constructed? Wow, what genome exactly is comprising of? So I asked about genome. So you mentioned a complete set of gene. If it is a complete set of gene, okay, we can call it as genetic information. Then we are studying on genetics. Genome, if you study on genome, we call it a genomics. If you study about the set of genome, oh, what do you mean by set there? What do you mean by set? I don't understand. Complete set of DNA. Still, where you get the DNA? What is the source of DNA or gene? Whatever you mentioned. Collection of genes. From where? Whether we collect from tissues, like we are collecting from heart tissue, liver tissues, where exactly we can collect the sample to sequence the genome. Because nowadays, if the people are approaching for diagnosis of rare diseases or even cancer diseases, most of the clinicians prescribe to go to come with a genome sequence. So earlier, to establish a genome, the first human genome established in 2003, the cost of the human genome is around $11 billion. Okay, it's around $11 billion. Okay, so if you find this much expensive, okay, now the cost of human genome is just $1,000 question, just $1,000. See the price. Okay, yeah, they've spent around $9 to $11 billion. So when they find it after it came around that much money. But now the human genome is just $1,000. It accounts around 86,000, isn't it? So even for the cancer patients, they spend a lot. So establishing a genome with 86,000 is made simpler, okay, for many of the patients, isn't it? So what exactly is a genome? Then if the patients are approaching the clinicians, how they go for characterizing the genome of a human. So actually, if you want to discuss on the genome, okay, the genome is actually developed. What is the shape of a genome? The next question is the shape of a genome. So I'll be answering for all these questions after this question. What is the shape of a genome? Anyone have an idea? Or can I take it for polling? What is the shape of a genome? Just answer what I know. Very good. One has answered. Swapnil has answered that whatever genes we get from gametes, but it's not just from the gametes. It's also from the other sources, other organelles also. Codon? No. Gametes are just the sex chromosomes, isn't it? Not just the sex chromosomes can sequence the genome. Then I think I have given a hint at present. Now itself I have given you a hint. It's not just the X and Y. Then what is that? Obviously, of course, mitochondria is also used. But the major thing, I just take you to the website and I can explain you further. NCP genome. Even I can take you to the Google pages. So first I can show the shape of the genome. Uh, Genome, human genome. All the genomes have see, even if you search in Google, it's difficult for me to find. Okay, I better take you to the NCP genome. And if you want to search for Homo sapiens, typing the species name. I'm just search. I have to search for a genome, isn't it? Genomes. Gone to the genomes or human. I can type either keyword like human or Homo sapiens, taking you to the. Okay, meanwhile I can see the answer. What else you have answered? Double helical. No one has answered right. Of course, chromosome is right. The genome is characterized from 22 autosomes. Only from haploid chromosomes only, the entire genome is sequenced. So obviously we know that we have 22 haploid chromosomes, okay, autosomes, and then we have X chromosome and we have Y chromosome. So totally 24. Okay, these 24 chromosomes comprise a circular genome. The shape of the genome is circular. So we have 24 chromosomes to make a circular genome. Also, the mitochondria, whatever we have a mitochondria, so even the mitochondria can also be used, okay, to sequence another genome that we call as a mitochondrial genome. We have a normal genome and a mitochondrial genome. So two genomes can be constructed from human. So if you want to see about the genomes, I am taking you to the website URL called ncba.nlm.nih.gov. Okay, so this is the URL I am taking you to for the genome. So this is actually, I think everyone know about this. Okay, NCBA stands for National Center for Biotechnology Information and National Library of Medicine, National Institute of Health, and government stands for US government. Okay, sir, 22 pairs or 22 chromosomes. We are not going to take pairs, we take only 22 haploid chromosomes, not the sets. We take 22 haploid and we take the gametes, that is X and Y. Okay, so totally it comprises 24 chromosomes. 24 chromosomes can map a circular genome. Okay, so now I just type a circular genome map. So this is how a genome looks like. If you want to see a genome, okay, the genome is mapped like this, like how you find a CD or DVD. So generally, if you try to write the content, it will be written initially. So actually, whatever you write, okay. So my pen is not working. Okay. So whatever you're going to write in the genome, okay, so it's going to write in the middle, and whatever you write in the CD, it will be in the gray region. And if you write much data, so much of the gray region you can find in the CD, isn't it? Similarly, so first after mapping the chromosome 1, maps the chromosome 2 and then chromosome 3. Like this, 24 chromosomes are mapped in an organized structure that is in a circular shape of a genome. So not only the human genome is circular, all the plant, bacteria, virus, and animals. So all the genomes are in circular in shape. Okay, and I'm browsing it due to the human genome. Okay, just taken it to the human genome. If you want to browse specifically, you can click the genome and you can focus on a particular gene. Like for example, if you want to know uh, what is the chromosome for insulin, you can search here. Okay, go to the genomes and you can type the insulin protein. You can find the information. Other than this, what else you can able to find? So the genomes are mapped through several sequences like ESTs, GSS, contigs. So there are different sequences which can map the circular genome. So the genome size of human is around 3.2 into 10 power 9 base pairs. 3.2 into 10 power 9 base pairs. If so, we have numerous content, isn't it? So nowadays, it takes just a week time, okay, or maximum of 10 days time from Illumina sequencer to sequence the entire human genome. So this is happening within a span of 7 to 10 days. So everyone can able to find the defect gene for the causative of a specific disease, can be cancer or any rare diseases. Okay, so this is something about genome. Okay, if you want to take further in the genomes, I'm just getting inside. And so if you go to the genome, obviously you can find, okay, so these genes are mapped inside a specific chromosome, isn't it? So one can also type a particular gene, or you can find a thing. Okay. So now we can able to find this information in the genome. So nearly can see this 22 haploid chromosomes and the X and Y chromosomes. So these chromosomes are mapping such a kind of a circular genome. My next question is how many genes, I mean, how much percentage of genes code for proteins in human genome? How much percentage? Very good. The answer is right. Less than 2 percentage. Okay. How much percentage of genes code for proteins in human genome? This is the question. But immediately I got an answer that is less than 2 percentage of genes. But if you see for yeast, you can find more than 99 percentage of genes, okay, code for proteins. But see, we have more than 98 percentage of genes ideal to code for any protein. In a nucleotide, you have two parts. One is coding gene, the other is non-coding gene. Coding gene is what we call it as codons, and non-coding gene is what we call it as exons, isn't it? So all these codons and exons, of course, can will be there in your human genome, which is taken from the chromosomes. And of course, the source what we take for uh, mapping the human genome, for characterizing the human genome, is the blood. They take a human blood sample, and they can characterize the chromosomes, and then of course they sequence all the genes from the chromosome, and then they map it to form a circular human genome. So, what are the essential characters present in DNA? So everyone know ACGT, but some programs also recognize this. For example, Perl program can also recognize these bases in few functions. But ACGT is what the characters we observe in the DNA as well as an RNA sequence. So whatever biological database is present, okay, even for RNA, you can only find ACGT. So you won't find U, okay. So only thing is that we can write programs and we can convert TSU. That's possible if there is a need. And if you want to know a few more information, one should know that we have ACGT for DNA or RNA, and for protein we have 20 characters, 20 alphabet characters, isn't it? So, what are the characters which not code for a proteins? One should know that. That is, according to the single letter characters. Okay, if you take the single letter characters which are not coding for the proteins, that only I can show. So obviously, you can understand that the other characters code for proteins. So B, there is a character called B, so which is not coding for any proteins. Then what else? Like P, we can have O, we can have Z, and then we can have. So if you take A stands for alanine, C stands for cysteine, T for aspartic acid, E for glutamic acid, F, G, H, I, J, J is not coding for any amino acid. K, L, M, N, O P Q or S T U. U is not coding. V W X. So these are the 6 characters which won't stand for any single letter amino acid. These 6 characters alone won't code for single letter amino acid. So the remaining 20 characters will comprise single letter amino acid. The remaining 20 characters code for proteins. So one should know about all this before going to check with the biological sequence databases and structure databases. So if you see the biological databases, so from nucleotide you can find GenBank, DDBJ, EMBL. So that is instead of taking in this form of a table, I can take you to the website and I can show how the sequence looks like and how can we retrieve the sequences. So to retrieve the sequences, especially nucleotide sequences, one can go to the NCBI website. So the URL of NCBI I already mentioned, ncbi.nlm.nih.gov. So then if I want to take a nucleotide sequence, okay, I can go to the nucleotide. So keep a nucleotide and search for a specific protein. So whatever you're going to search, okay, so whatever you are trying here, this we call it as enters. This is an interface called enters in NCBI where we can search for data in the databases and we can also use the tools. If you want to use any tools, we can also use the tools from here. Okay, now I'm presently checking a nucleotide sequence called insulin. So taking you in to take an insulin nucleotide. So if I search for nucleotide, you can find mRNA, you can find linear DNA, you can also find circular DNA. So what are the sequences I can get? Linear mRNA, again I'm telling you, linear DNA as well as circular DNA. So here at in the first page you don't find, yeah, but you can also find circular DNA. So these are the information you can find. Okay, if you want to find specifically, okay, an mRNA alone, one can click this. So this is an advanced filter. If you want the Homo sapiens alone, you can click this. This is another advanced filter. So now for initially I'm clicking the Homo sapiens. We can able to find 11,518 sequences out there. So these are the total hits for insulin. Okay. If you want to go for specifically ESTs, you can click EST. What are ESTs? I already told you. Express sequence tags. Of course, the sample is nothing but an mRNA, a linear mRNA. If you want to take the mRNA sequence, you can use any of these formats. The common universal format, what we can use is FASTA. The FASTA is the format, is the universal format, which is present in almost all the biological databases. So this is the largest database. You know, in NCBI, GenBank, you can find more than 300 million nucleotide sequences in 2010. But now I don't know. The size is increased rapidly, isn't it? So we have almost touching billions of nucleotide sequences deposited here. So whatever even you are searching, okay, you are searching with this billions of sequences, nearly millions to billions of sequences. Now, if I go to the FASTA format, if I want to get inside, I'm going to click this FASTA hyperlink. After clicking this FASTA hyperlink, you can able to find the description of it. And the first line generally tells you about the identifiers, database identifiers of the sequence. That is, whatever you find initially, so this is GenBank accession number. And whatever you find, this is a keyword, which we call it as locus name. Also, the species and whatever they mentioned or comes under locus name. So if like how we can identify a student using a roll number, registration number, name with initial. Similarly, the database sequences can be recognized using database identifiers. This is a NCBI GenBank accession number. So If you want to search this sequence, if you know this accession number, you just copy this and you paste it instead of typing insulin, going to the Homo sapiens. And again, I'm selecting an EST database. So plenty of filters we're using, isn't it? If you know the accession number, simply type it to get this kind of a sequence. So I told you in the first line, generally you find the information of the sequence. Just because we can't able to see if the sequence length is more, okay, we have a wrapping of text, wrapping. Okay, nearly you have 60 characters in each and every line. So after 60 characters, 61 to 120 is present in the next line. Okay, so for a user friendly, like for to user friendly effect, so they are wrapping the text, but generally you have to remember in faster sequence, you have the first line as the identifiers of the particular protein or particular DNA or RNA, whatever you're searching, and the second line is full of the sequence. If it is an RNA, you find ACGT. If it is a protein sequence, you find the 20 alphabet characters, other than whatever characters I mentioned, which we can't find that. Okay. So this is something about how you can able to find the sequence from the nucleotide database. Okay. And like nucleotide, we have plenty of sequences. Like if you want to search for genome, so initially I tried with genome, isn't it? So if you want to check for a gene, you can click gene and type insulin. So if anyone didn't try, so even if you're in first year or second year BTech or BScs, so the UG graduates can try. Okay, first open the NCBA and try with each and every database, and we just type a common protein name like a insulin or myoglobin, whatever you know. You type the protein name alone. Okay, and so you can also find plenty of sequences. The most popular what we use is the nucleotide and the protein. Inside the protein, I am going to type now insulin. So till now, uh, I just fetched you an EST sequence for an insulin protein, isn't it? Now I'm going to fetch you the protein insulin of Homo sapiens. Number of hits what we can find from Homo sapiens is 7,141. There are 7,141 protein sequences of Homo sapiens. But you can able to find these sequences. Why we have this much sequences? You know, in human, that is Homo sapiens, you have only two proteins. One is insulin ligand, the other is insulin receptor. So whatever the small size you can find, 110 amino acid is the insulin ligand. So this is the total size of the insulin ligand. Insulin receptor size is more. Then what is this isoform and why we have plenty of insulins? One is 110, 107, 94. So if you're doing a project with a diabetes, and of course you want to take this protein sequence, insulin protein sequence, just use this 110 amino acid. Okay, don't use this 107 because few amino acids are deleted here. You know, the insulin can also be engineered. We have find so much recombinant insulins in our market, isn't it? So if you want to take a while and naive insulin protein, better go with this. So how we can able to find whether the insulin ligand is just 110 amino acid? So for this, we can also go to a database called Uniprot. There is a database called Uniprot. The NCP is maintained from US government. And Uniprot is majorly from UK. But it's also associated with two more countries, Switzerland and Germany. Okay, it's also associated with two more countries. Now, if you see this Uniprot, there are plenty of databases, like how I showed you protein nucleotide from NCBI. You know, whatever I showed you as a nucleotide in NCBI, we call that as GenBank. And whatever protein sequence I just searched in NCBI, we call that as GenPept. GenBank, GenPept. Okay. And here in Uniprot, if you want to search a kind of a protein sequence, generally you can go with Uniprot KB, that is Universal Protein Knowledge Base. universal protein knowledge base. Here, I'm going to search for insulin. Like similarly, how I searched insulin in NCBA. So after searching, I got numerous hits, nearly 3 lakh hits. But if you see the Homo sapiens insulin ligand, okay, we have only one. What is the next then? Next is the insulin receptor, INSR. INS default mean insulin ligand, and this is insulin receptor. Got it. So we can able to conclude this, and not only can conclude even the sizes of the protein of the naive proteins, but also the detailed annotation of a protein we can fetch from this UniProt. So before taking you to these hyperlinks, okay, these hyperlinks are the UniProt ID entries. UniProt ID entries. Generally, the UniProt ID is a 6-letter alphanumeric letters. You just start with P and followed by 5-digit numbers. Like how I told you NCBH and bank accession number and locus names. Here, if you want to fetch a kind of a human insulin ligand, one can type this entry. If you type the entry here and click for search, you get only this entry alone. So you can get more information on this by getting inside the hyperlink. But before clicking this hyperlink, I take you to this NCBA GenPept database. Okay, so you can find GenPept here, isn't it? Earlier in a nucleotide, you can find GenPact. Okay, so okay. Now, if I want to take you to the FASTA format, click this FASTA. So here in the FASTA format, you can able to see the characters, a single letter amino acid characters. So this will be the second line, and then the first line you can find the identifiers like GenPept accession number, and this is of course whatever the protein name and the species name or the locus names and we also have a kind of a GenPank accession number for even though we searched a GenPept database, even for proteins, they call this as a GenPank accession number. It is a GenPept database, no doubt in that. But like DNA and RNA, here also you have a GenPank accession number, okay, which is common to all the sequences. Okay, so just a few information on they are getting, just the sequence alone, isn't it? And if you go to the GenPept, that is this in another format of we can find in NCBA, you get some information like a partial annotation, but not a detailed information. Like you get a length, you get like a linear protein sequence, and you get something like uh, what is the taxonomy? It's from Homo sapiens, but what is the taxonomy? That is from eukaryotes, we can find Homo sapiens so much branches in taxonomy can find. You can find the authors who has first sequenced the protein and deposited in the NCBA. And not only that, you get who is the first person, who are the scientists published this protein in a journal. This is the journal they published in 1980, and the PubMed area of the journal is this. Okay, so you get these information and additional information, but whether these information is we can use it for any project? No, isn't it? Then what exactly we need for doing projects? If you're layman, if only if you're stepping freshly in bioinformatics and want to do some projects in bioinformatics, one should know you have to use this database called UniProt KB for full annotation. Now I'm clicking the first hyperlink for insulin ligand. Now I'm retrieving an insulin ligand protein. So you can find so much information about the protein. Names and taxonomy, subcellular location, disease. So whether is it associated with the disease, and how about the post-translational modification, expressions, interactions, whether insulin ligand is interacting with the receptor, insulin receptor alone, or it's also interacting with other receptors. And how about the structure? What are the 3D structure of the insulin? Where can we download this 3D structure? We have hyperlinks for this. And not only that, we also have secondary databases associated with it, and we also have family and domain databases linked with it, sequence information, isoforms, etc. And what is this isoform? The isoform we can also see here, isn't it? So when I'm searching this, okay, not only we are getting this insulin, but we are also getting something like isoform. If during the sequencing, okay, you have a trouble, like your sequencer is not giving a high quality reads. If your sequencer is not giving a high quality reads, and whatever is you can able to sequence with your sequencers, even that you can take it for depositing in the NCTA. They don't mind at all. So why we have this much number of sequences? We have only one protein sequence, but we have this much 7141. It means that a person from Chennai, if he is sequencing at present, okay, and if you get a kind of an precursor, like an isoform precursor, he can deposit. And another person from Bengaluru, okay, he is also sequencing it and is getting something like a 94 amino acid, like a precursor, like a partial protein, he can also deposit. On the same day, we may get some multiple sequences, some 10 to some, we can also get some 10 sequences. So now we've got 7141. If you search after 1 month or 2 months, even the size will be increased. The number of hits will be increased. But in Uniprot, it's not like that. We don't have redundant entries. We have only one entry. We have only one entry for a ligand protein as well as for a receptor protein. So it's easier for you to connote even the information. Okay, so if you see this insulin human, okay, just drag down to find the plenty of information. So you can also find the binding sites. You can also find each and every specific motif of the protein. Like what is a functional protein which gives the function of an insulin? You know, the insulin is used for regulating the blood glucose level, isn't it? So not all the 110 amino acid is going to regulate the blood glucose level. Only a specific part of a protein, around I think 93 to or 97 to 103, something like that. So, a few amino acids, only the few amino acids, okay, which going to constitute an insulin motif, that is going to regulate the blood glucose level. So what are the sequences? What are the motif part, and how about the pathways associated with the protein? What are is it linked with the enzymes? So what are the enzymes? So all this information you can find. And related to proteome, where can we find even in genome? You know, the insulin is present in chromosome 11, insulin protein. So also, if you want to dive to the proteome, okay, we have a proteome identifier. If you click this, it take you to the proteome. Then what is a proteome? We just discussed about the genome, and we discussed about the shape and size of the genome. What is the proteome? And even I asked you to define genome, isn't it? Genome you can define as, of course, you have a complete set of genes, but it is organized. It is organized. It's actually complete set of genes isolated from haploid chromosomes as well as the gametes, and we organize it in the form of a circular shape. How about proteomes? The proteomes don't have any shape. Whatever family of proteins, okay, you have done in the sequencing, you have got from the peptide mass fingerprinting, you have done in the 2D gel electrophoresis, that you take it as an proteomes, a cluster of identical proteins. Okay, so that's what a proteome. We don't have a defined shape and size for a proteome. If you see the other information, what exactly can find in the Uniprot KB, we can find whether this is involved in disease or not. Yes, this insulin ligand is involved in hyperproinsulinemia disease. About the disease and what are the different binding sites which are most important for this disease, you can get this information. Not only that, we can also have a link with the publications. See, Much information is there, isn't it? About the second disease called type 1 diabetes mellitus, and even for a variant called 2, T1, T2, and for this, and PNDM4, and we have MODY10. So we can find four different diseases. Our insulin ligand is linked with four important diseases that were represented here in UniProt database. And then we can also find uh, the natural variants. We can find organism specific, like if you want to find a disease gene networks, this is a special database. Disease gene networks. Okay, if you want to know about your insulin is linked with what kind of a diseases and how about the genetic variation. So one can go with the DGNet. Okay. So now I'm taking you to the DGNet. I'm going to click this non-insulin dependent. Okay. I'm taking to a specific disease gene networks. So we have types of networks like gene-gene interaction networks, protein-protein interaction networks, gene transduction networks, and also disease gene networks. This is a disease gene networks. How a disease can also link with other diseases. That also we can able to find. Okay. And how exactly it is linked. So more information you can find in this gene network. Also publications are linked in the bottom. You can find in detail about that. Okay. So this is something about the gene network. Then what else we can able to find? We can also find few more databases linked with pharma or for net open targets. This is for finding the binding sites and even finding the appropriate ligands. For finding the appropriate ligands, we can also link with the CHEMBL. You know, for controlling the insulin level, you can go with the ligands like glycoxide, empresol, myristic acid. If you want to go to this drug which is regulating the insulin, you click this. I'm just clicking this. This is the ID, a drug bank ID of myristic acid. If you want to check the information of myristic acid, scroll down. Here also you can find much information of the drug. This is also a fully annotated database for drugs. Okay. Like you can find the pharmacology, you can find the interactions, products, categories. If you want to purchase the drugs, you can also find the vendors, chemical identifiers, references, who are the various scientists worked research on this myristic acid compound. What are the clinical trials completed on what kind of a diseases, pharmacoeconomics, targets, a target towards specific receptors. So all this information you can find from the drug bank. You remember yesterday I was explaining you about cheminformatics databases. What such a database is a drug bank. Okay. Now you can find it's our Uniprot website is linking with the drug bank. I just taken you to inside the drug bank. Likewise, you have so many databases or link in this Uniprot. Okay, these are the motifs, proteomic databases, post-translation database, gene expression, okay, binding sites, even 3D structure of proteins, protein-protein interaction databases. I can take you to the one database. This is BioGrid. The BioGrid is a protein-protein interaction database. So if you're saying the insulin ligand is interacting with these proteins. Nearly it is interacting with 528 proteins. So more about this, one can go click here inside and you can go to the network, protein-protein interaction networks. Okay. So more information you can find even in other databases like DIP, string. If you click the string, you can find much information. That is the insulin growth factor 1. The insulin ligand is linking with the IGF-1, IRS-1, EGF, IDE. So the top 50 proteins uh, interaction with insulin is shown in this string database. This is a protein-protein interaction database. Okay. Likewise, you can find numerous, like even the 3D structure of a protein, how a 3D structure of a protein looks like. This is the 3D structure of our insulin protein. Okay, so one can able to see this structure, and if you want to download this, use this. If you want to download X-ray crystallography generated 3D structure, you can use this PDB ID. This the PDB ID is generally 4-letter ID, alphanumeric letters. If you want to download the protein structure, click this. It can take it to the PDB website. Okay, or you can simply download from here. Or if you want to go to the PDB website, where you can download the 3D structure of the insulin protein, ligand protein, click here. Now it's directing to that. Okay, so this is a human insulin mutant. Okay, we also have human insulin 9, but this is a mutant. Okay, you can see more information by scrolling down. Okay, so this is about the sequence length. There are 2 chains, A and C chain. Okay, so if you want to download the structure of the human insulin, okay, one can go here. Okay, so you can click download files. You can also download the FASTA sequence, but if you want to download the 3D structure of a protein, you can click PDB format. So PDB format, okay, I'm going to save it in bioinformatics demo. That is 1B insulin ligand. I'm saving this protein like this in a desktop. Now I'm opening this desktop folder. If I want to open this protein, I have installed Discovery Studio Visualizer. You have to install a protein structure visualizer to visualize the 3D structure of a protein. Of course, whatever you're viewing here can also explore here in 3D in online. But if you want to download and visualize in offline, that is also possible with that. So this when I clicked in online, so this is the structure I can able to find for this protein. The same in Discovery Studio Visualizer, no offline software. This is a freeware. Anyone can install this. Go for the Discovery Studio and download that software, and one can see our insulin protein. Okay, this is how a protein looks like. So we can zoom in, we can zoom out, we can interact whatever is possible with interaction. Okay, so this is something about UniProt, the PDB for the database structure, and even for in NCBI, even in NCBI, I just close, which is not in use. So even in NCBI, you can download the 3D structure. So what is the 3D structure we found? The PDB ID, if you remember, we can type that. For that, you have to go to the structure. Inside the structure, instead of typing an insulin and searching specifically, we know that 1B9E is the PDB ID, isn't it? So if you type 1B9E in the structure, it takes you to the MMTB database. In NCBI, we have a database called Molecular Modeling Database. There you can find the 3D structure of a protein. So this is the structure of the insulin protein. If you want to download this. In what format? So you have different formats like ASN.1, PDB, XML, JSON, image. If you want to download in 3D structure, select the PDB and then you click download. Got it? So you can also find the PDB structure in NCBI MMTB database, which is meant for structure. Okay. So till now, what we have discussed. So till now, if you see, just a recap. In nucleotide sequence, we discussed on GenBank, and protein sequence we discussed on GenPept, and we also discussed something about the structures, about the PDB and MMTB, and this is the extract I've taken from a textbook called Bioinformatics Computing by Birkinon. One can look through that also. Okay, and what else did we discuss? We discussed about protein-protein interaction databases. Okay, and we have seen that is gene networks. Okay, so this information I can learn from the textbook, so I didn't show that. So I'm taking you to this slide now. So we have enzyme database. I already showed you a disease database like the GNet, and of course, OM also comes under that. OMIM stands for Online Mendelian Inheritance in Man, and chemical database, microarray database. So these are all present in NCBI itself. Taxonomic database. Let us complete this slide alone today. Okay. So what exactly are the biological databases we can find from NCBI? So can we go for now chemical database and the other databases? So I take you to the NCBI at present. So in NCBI, okay, if you want to go for a chemical compound, okay, like myristic acid, this is what we have seen recently, spelling myristic, or I can go for aspirin, a popular drug, acetyl salicylic acid. You can type even paracetamol. Instead of paracetamol, you can type the generic name of the paracetamol, that is acetaminophen. Then you can find the structure of the paracetamol. Now I'm searching for aspirin, acetyl salicylic acid. If you want to get inside the aspirin, click this link. Get inside the aspirin. This is what found in the NCBI, National Library of Medicine. We can download both 2D and 3D structures. For downloading the 2D structure, scroll down. In 2D structure, you have download coordinates. Click here, save as STF format. But generally, we use the 3D structure. So instead of 2D structure, you can go for 3D structure and download the 3D structure STF format. This is what we use for docking, simulation, etc. So more about that chemical compound, you can find, if you can scroll down, you can find all this information like chemical physical properties, spectral information, related records, chemical vendors, pharmacology. So much of the information you can find by scrolling down in NCBI PubChem database. Like in NCBI PubChem, the popular database, what else we can use is PubMed. So PubMed is generally used for downloading the biotechnology, that is PubMed is for biotechnology literatures. PubMed, if it is for biotechnology, there is also another database called Medline for clinical literatures. If you want to download clinical literatures, one can go with the Medline. Of course, these two are associated. And if you see this PubMed, I just typed aspirin. So whatever papers published related to aspirin, you can able to find. We can able to find around 76,188 hits. Okay. So this is what we can get some information from the pub. So the most important databases we have searched and related to microarrays, one can search in GEO datasets and GEO profile. So this you can use it for downloading. And for disease related, one can go with OMIM. You can go to the OMIM and type any disease name or type any specific gene responsible for it. If I type asthma, you can find this information. One can search the information of genetic related the asthma disease through this information, through this hyperlinks. Okay, so we have discussed all these things. So except metabolic database. So for metabolic database, one has to go to a popular database called KEC. So this is the last database I'm going to show it now today. That is the most important database for metabolic pathways is Kyoto Encyclopedia of Genes and Genomes. Kyoto Encyclopedia of Genes and Genomes. Inside that, okay, if you want to go for a pathway database, I'm going to click pathway. In KEC, we have different databases like pathway, genes, enzyme. Like go back and show you what else you have. So pathway, genes, genome, compound, enzyme, reaction, network, disease. So these are the popular databases present in Kyoto Encyclopedia of Genes and Genomes. This is majorly taken care by Japan government. Okay, so now you find .jp, genome.jp/KEC. So now I'm clicking KEC pathway database. If you are focusing on a specific disease called asthma, so other than a disease, you can also search for metabolism pathways like glycolysis, citric acid, glyconeogenesis, gluconeogenesis, all this, whatever you studied in metabolism, like lipid metabolism, peptide, protein metabolism, all these things, amino acid metabolism. So you can find in metabolism database. We can also find for human diseases, organismal cellular processes. For all these categories, you can able to find. Now I'm just typing the asthma, a name of a disease. I'm going to search for that. So you can find a large pathway. This is a pathway. I'm just clicking that image. You are getting a large image at present. So this is the image you can get, and you can find what are the proteins linked in the pathway for the asthma. Okay. So that's it. I have completed my part on basic introduction of various biological databases. If you have any doubts, you can ask me at present. I have completed my lecture on today. So tomorrow we'll be discussing on data file formats, more about metabolic pathway, motif and domain databases, and whatever left out on today, we'll be discussing it on tomorrow. Any other questions? Sir, how we retrieve SP gene sequence from whole genome? So one has to go to the genome and we have to type that SP gene. O codes for pyrrolizing 22nd amino acid. Actually, these we just can able to find 20 essential amino acid characters. We can't find 21, 22, these amino acids, ornithine, all these things we won't find. Insulin protein is made of 51 amino acids, and why should we use? No, actually, insulin protein, I told you already, we have a ligand, we have a receptor, we also have insulin growth factor receptor, that is another protein. We have categories of protein. The insulin protein, you can confirm from the Uniprot. Okay. Any other doubts you have, participants? Okay. So thank you very much for your interaction. Okay. So we see you on the next class on tomorrow.